Okay, I also have to use the screen because your friend needs to participate. So, shh. Now, we are still in the first part of the graph. I don't want any side talks. We are still in the first part of the graph. Is your friend even there? Where is she? Excuse me?
One participant. So
I know. Where is she? Where did she go? No, you stay where you are. Complete waste of time. Go to whatever you want. Okay, enough waste of time. Now we are still here. We are still in this part of the graph. Okay, now we want to ask a very important question. How are the enzymes affected when the temperature is very low, when it's cold? Will the enzymes be denatured? No, never. Never. The enzymes are never denatured when it's cold. The only thing that you will mention that the enzymes will work slowly. They slow down. Why? Because less. Very good. Everything will go in the opposite direction. Because at low temperature, you have low kinetic energy. They move slowly, less speed, less chances of successful collisions, less formation of enzyme substrate complex, slower reaction, less products. Everything will just go in the opposite direction. Okay, usually the question is at A, asking you about what is happening to the enzyme at A and what is happening at B. Now at B, describe. First, we will describe and then we will explain because these two have different answers. If you want to describe B, what will you say? Very good. Exactly. When you describe, you have to go to the graph. I have to describe what is happening at the x-axis and what is happening to the y-axis. What is at the x-axis? Temperature. So at B, when I come to describe, this is exactly what I will write. As the temperature is increasing, the rate of the reaction is decreasing. This is the first mark. The second mark will go for what? Numbers. I have to give numbers. For example, when the temperature was 50, the rate of the reaction was here. When the temperature was 55, the rate of the reaction decreased, it became here. This is the second mark. This is describe, not explain. Explain, you will say something different. In explain, you will mention why. Why did the rate of the reaction decrease? What will you say here? Exactly. Because the enzymes will denature. And then still it is not enough, especially for year 10. When we come to describe or mention denature, we have to explain what is denaturing of the enzymes. What happens to the enzyme when they denature?
The shape of the enzyme changes.
Excellent.
So the bacteria cannot get into the active side and it's not complementary.
Perfect. Okay. When we come to mention the word denature, anytime you mention the word denature, You have to explain what happens when the enzyme denature. When the enzymes denature, what happens to the bonds between the amino acids? They break. So this is the first sentence. When you come to answer essay questions, clear, simple English. You don't have to complicate things. Line, line, line. The bonds break. The shape of the enzyme will change. The active site, its shape will change. Will it be now complementary to the substrate? No, it will not be complementary. It will not fit. So the substrate will not be able to fit. No enzyme substrate complex. No reaction. The reaction will start to slow down until it finally stops. Clear? Okay, so this is a very easy graph, and they like to ask questions, and it is nice if they ask questions because it's like a present. You will be easily be able to answer it. Yes. You don't have to use those terms in this graph at all. If you want to, you can say that at the beginning, as the temperature is increasing, the rate is increasing, and they are directly proportional. You want to use it, fine. If it confuses you, it's not necessary. Okay, so again, you have this table, this graph, really important. Temperature on the x-axis and the rate of the reaction on the y-axis. We will divide the graph into three parts, part A and B and C. At A, what's your name? Hala, my dear. Describe what is happening in A. Good, 1 mark. The second mark, I'll give example. Now explain why when the temperature is increasing, the rate of the reaction is increasing. Yes. Yes. Excellent. Excellent. All these are keywords that you will explain in this part of the graph. Enzyme substrate complexes, more enzyme substrate complexes, more products, faster reaction. B, optimum temperature. C, it's not enough to say it will denature. Yes, it will denature, but this is not enough anymore. It will denature because the bonds will break, because the shape will change, because the active site will not fit, etc., etc., etc. You will get the full mark. Clear? Good. Now, we want to actually do the experiment. Your friend is still not online. So, again, what is our reaction? Do I switch off the lights? Can you switch this one off, please? So, our experiment. We want to study the effect of temperature on reaction rates. Pay attention with me. Don't sleep, okay? So what will we do? Pay attention. I will get 2 test tubes. One test tube will contain the amylase enzyme, and the other test tube will contain the starch. And how will I change the temperature of these 2 test tubes? Very good. I will put them in different water baths. This is the water bath. And I will give examples. One time it will be cold, one time it will be warm, one time it will be hot. I will put these 2 test tubes in the water bath and I will leave them for 10 minutes. Why? Very good. So imagine with me, I have 2 test tubes and I will put them in the different water bath. It's very hot. I want them to be hot like the surrounding water. So I will have to leave them for 10 minutes so that they take the temperature of the surrounding water. How do we write this down? We will leave them for 10 minutes. If you want to say to take the temperature of the surrounding water, it's correct. It's very good to use this term to reach equilibrium. And I don't have to explain it further. Equilibrium means to take the temperature of the surrounding water. After I leave them for 10 minutes and they take the temperature of the surrounding water, now I will mix them. I will mix them and I will put them back in the water bath. So now what do I have? I have only 1 test tube in the water bath that contains the enzyme amylase plus the starch. This amylase enzyme will break down the starch to give me what? Very good. So the amylase enzyme will break down the starch to the maltose. How do I know that the reaction took place? What do I do? Think. You are correct. Okay, you said iodine. Do I add the iodine directly to my test tube?
No.
No, because then I will destroy my mixture. What do I do? Every minute, pay attention. Let me change color. Every minute, I will take one drop out of my mixture. So I will take out one drop every minute. And I will put this drop of mixture that contains starch plus the enzyme on a spotting tile. Where is my spotting tile? What is my spotting tile? 1 minute. Here. This is my spotting tile. This is my spotting tile. What is my spotting tile? My spotting tile is like a small plate. It's like a plate that I have in the lab that contains many holes. These are called holes or wells. And in every hole, I have one drop of iodine. What is the original color for the iodine? Orange brown. Okay, now I have my mixture. This mixture is the enzyme plus the starch, and it is in my water bath so that the temperature does not change. Every minute, I will take one drop out and I will put it on my iodine. If this mixture contains starch, what will the color change? It will become blue-black. Okay, let me change the color. This will be blue-black. This means that the starch is broken down or not yet? Not yet. Very good. After another minute, I will get out another drop from my mixture and I'll put it in the second well. And I will look at the color. If the color changes to blue-black, this means starch is broken down or not yet? Not yet. I will keep on repeating this step until the iodine changes color or remains yellow-brown, remains yellow-brown. So I will keep on repeating this step until the color, very good, remains yellow-brown or doesn't change. If this happened at the third well or after 3 minutes, so how much time was needed for the starch to be completely broken down? 3 minutes. So it took me 3 minutes for the starch to break down. Clear? Very good. Now, the shorter the time, the faster or the longer the reaction? The faster the reaction. Okay, I want you. انا في الزوم اهو والله شغاله وبعت لها لينك تاني كمان على الواتساب. ما اعرفش انا شغاله بس انا مش عارفه، انا ما اعتقدش دخلت. هو one participant. انا بس انا مش عارفه ما دخلتش ليه. وخليها تبص على الواتساب جروب بتاع الكلاس، بعت لها اللينك. Okay, now we will watch this video, please. If you did not understand the experiment, hopefully this will help.
Label 6 test tubes A to F. Add 1 cubic centimeter of amylase solution to each tube. Label another 6 test tubes 1 to 6. Add 5 cubic centimeters of starch solution to each tube. Group tube A with tube 1.
These are the water baths.
Tube B with tube 2, and so on. Put the test tubes into beakers of water at different temperatures. Leave the tubes in the beakers for 10 minutes.
Why?
For each beaker, start the reaction by pouring the starch solution into the amylase solution. Shake the mixture well. Start the stopwatch immediately and record the time as 0.
This is the spotting time.
Transfer a drop of each reaction mixture to an iodine drop on the stop plate with a clean dropper. Stop when the iodine drop remains brown. This is the time at which all the starch in the mixture is broken down.
See, the last drop,
Record the time.
the color will not change.
Present the results in a table.
Okay. Is this experiment clear?
Yes.
Good, very good. Now they will ask you, what are the sources of error? What are the sources of error for this experiment?
So basic for the water. The temperature might be.
1 minute. Let me just open a screen. Yeah, so now the experiment does not end here. Did you notice the table? Did you notice that the table, the independent variable was on the left side? What was the independent variable? The temperature, independent, the temperature, yeah? I'll write the temperature and degrees Celsius up there, the units in the header. I never repeat the units in the body of the table. What will be put on the right side of the table? The time taken for the iodine to remain orange-brown or not to change color, and in minutes. Good? Now, the common question, sources of error. What are the sources of error? How to improve? Very important. If you will write this down, it will really help you with paper 6. So the sources of error, tell me.
There was no insulator to water.
Okay, if you notice the experiment by this scientist, he used a normal beaker. So will the temperature remain constant or it will fluctuate throughout the experiment? Okay, so the temperature, yes, he did change it at every beaker. Exactly. But the temperature can change throughout the experiment. So how do you improve? Very good. A thermostatically controlled water bath. And I think you know this by now. It's a water bath connected to a socket. The temperature of the water in this thermostatically controlled water bath does not change. Give me another error. What's your name? Yes. Hey, my dear. Give me another error in this experiment. What were the results? Did the results involve a color change or not? Do you understand the experiment? Okay, didn't the results involve a color change? Don't we see if the iodine will be blue, black or not? Yes. Isn't this an error? Didn't we say many times that any result that involves color change? Very good. Haya, are you with me, my dear? Didn't we look at the color change? Is this accurate? Is this accurate? What if I do not see the color very well? What if I argue with you? No, 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 it changed. No, 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 it did not change. Is this accurate or not accurate? Huh? Not accurate. So any experiment that involves a color change, the color change in itself is inaccurate. The color change in itself is an error. Hiya, my dear, how do I prevent this mistake or how do I improve it? Which device do I use to change the color to a number? Hiya, we said it many times, my dear. We use a device called the colorimeter that will change the color to a number. So I don't argue and I don't do mistakes. Okay, you two girls back there, are you paying attention? Okay, yes. Another error. Think of errors. Exactly, exactly. He did all the water baths at the same time. Each one separately. Perfect. So the starting time for the reaction will be different for every experiment because he did them all at the same time. He mixed and then he put. He mixed and then he put. He has to do them all at the same time. He cannot do it all at the same time, or there has to be one person for every beaker. So how do we improve this mistake? Do every experiment separately. Usually they ask for 1 or 2 errors, but we want to think of all possible errors because they can ask you for 3 if they want to give you a hard time. Another error. What about the dropper? Exactly. The dropper, he has to use a different dropper every time so that the solution inside the dropper is not diluted by the previous solution. And how do you know that the drops are the same size? Okay, so using a dropper also is a source of error because the size of the drop can be different every time. So what do we use to improve this mistake? Prevented, pipette, burette, blah, blah, blah. So put it a rule of thumb. Anytime there is a color in the experiment, this is a source of error. Anytime there is a dropper, also this is not accurate and it is a source of error. Clear? Excuse me? No repetition, no control. No one talked to me about the control. What will be the control in this experiment? And do not tell me room temperature.
It starts without adding amylase.
Excellent. Okay, again, rule of thumb, write it down. Write it down. We said very important points today. Any experiment with color is an error. Any experiment that uses a dropper is an error because I can never confirm that the drops are the same size. Girls, are you paying attention with me? Is my voice clear? Is everything clear? And we said something also very important. Any experiment that we will ever face from now on till the end of the year that involves enzymes, the control will be, I will repeat the same experiment, but I will boil the enzyme. Or I will simply don't add the enzyme. Yes. So that I'm sure that the color change is the result of the enzyme being present. When it is dead, it will not cause a color change. When it is not there, it will not cause a color change. So I either boil it or remove it or replace it with water. But do not ever say that I will use the room temperature as a control experiment. The room temperature can be 25 degrees Celsius, and this in itself can be one of the temperatures that you are using, or close to one of them. So it can never be a control. Clear? Yes, my dear. Quickly. Hiya, my dear, do you understand the experiment? Sure? Okay, so going back to my investigation, we want to study the effect of temperature on amylase. This question is very common in paper 6. It nearly happens every year. This question, the investigation question, out of 6 marks. Now I hope that we can answer. How do we answer this? Where do the 6 marks go? Wait, where do the 6 marks go? Excellent. So one by one. The independent variable, what will it be here? Excellent. Please write change in temperature or different temperature to show the examiner that I will change the temperature. What is the second mark? Excellent. What's the dependent variable? Remember the experiment. What were we looking at? We are waiting for what? Very good. So the time. So the dependent variable will be the time for the iodine to what? To remain yellow-brown or to change to blue-black? To remain yellow-brown. It will change to blue-black if starch is present. We want to know if starch has been completely broken down. So we will keep on adding iodine until the color stays yellow-brown and never changes to blue-black, like you saw in the experiment. Okay, so pay attention to these details. So the dependent variable will be the time needed. Oh, God. So again, this is so important. The independent variable will be the change in temperature. The dependent variable will be the time taken for the iodine to remain yellow-brown, not to change to blue-black this time. Okay? Understood for everyone? Now tell me, what can be the constant factors? Very good. What is the word that I will use here? Same. At least 3 sames. Same what? Same pH. Give me another same. Very good. Same volume of enzyme. Same volume and concentration of starch. Same volume and concentration of starch. Very good. Same iodine, same volume of iodine in the spotting time. Excellent. Now the steps. Do you understand the experiment? So now you will be able to write your steps clearly and simply. You don't have to write so much details and waste your time. So if I am you, I will say what? I will get 2 test tubes, 1 test tube with the starch, 1 test tube with the amylase. The first sentence. I will put these 2 test tubes in what? Different water baths and give examples. Cold, warm, hot. I will leave the 2 test tubes for to reach. Very good. After 10 minutes, I mix. Every minute, I take 1 drop out, put it on the iodine in the spotting tile. And keep on doing this until the color of the iodine remains yellow-brown. I finished. I finished the steps in 3-4 sentences and I got my 2 marks. The last 2 sentences that I throw them in any investigation. Repeat and calculate average. Repeat at each temperature and calculate average. And for safety, I wear gloves. The water bath in itself is a safety precaution. Clear? So after the steps, I will say repeat at each temperature and calculate average. And I have to protect myself. Clear? Any questions? Good. I don't know where your friend is up to now. Okay, so then I can use the board. Good. Let me use this side of the board. Now, there is another investigation that is also very common, which asks you to study the effect of pH this time on amylase. Those who solved paper 6 last year know how common this question is. Okay, so again, let's solve it together step by step so that we don't lose the 6 marks. What is here the independent variable? Excellent. So here I am going to change in pH. What is my dependent variable? Again, the same dependent variable. The time taken, very good, for the iodine to yellow brown. Excellent. To remain yellow brown. Now, the constant factor. Yes, very good. This time we will keep the same. Excellent. The same temperature, and everything else will be the same. Same volume and concentration of enzyme, same volume and concentration of starch, same instruments, same type of iodine or same dropper, same volume of iodine and the spotting tile. Girls, please pay attention. Please. Read them. Okay? Now what did I do in this experiment? Again, I will get 2 test tubes also. I will put my starch in 1 test tube. And I will put my enzyme, which is the amylase, in another test. With the enzyme, I will also add something called a pH buffer solution. Now, before you ask, what is the pH buffer solution? It is a solution that is ready-made in the lab. I already have it in the lab of different pH. I have pH buffer with pH 2, pH 7, pH 9, pH 10, pH 11. Okay, and I changing the pH. I changing the pH. So I will add here the enzyme, plus I will add some of the pH of the solution. So now the pH here of the enzyme will change. After I change the pH of the enzyme, now I will mix. So now I have a single test tube that contains the starch and the amylase and the what and the buffer with a particular pH. That's the pH 2. What do I do next?
Wait for 10 minutes to each.
No, we don't do this here. It's not a water bath.
Oh,
It's not a water bath. Okay. If you want to fix the temperature, we can put it in a water bath so that it stays constant. But we're not here involving the temperature. Okay. Don't get confused. Every minute, I will get out exactly like we did before. I will get out 1 drop and put it on iodine in a spotting tile. And I will keep on repeating this step until the color of the iodine remains yellow brown. Wait, let me finish. So I don't want you to be confused like your friend was a minute ago. In the first experiment, when we were studying the effect of temperature, in both experiments, we will start with 2 test tubes. When we were studying the effect of temperature, we put the enzyme in 1 test tube and the starch in another test tube, and then these two in a what? Water bath. We wait for 10 minutes and then we mix. Okay? In the second experiment, when we are changing the pH, we don't need any water baths. And we will not wait for 10 minutes until we reach equilibrium. No. We will also start, yes, with two test tubes. One with the enzyme, one with the starch. But in the enzyme, I will add something. What do I add? pH buffer solution of different pH. One of you will ask me, can't I add a different acid or base? Yes, you can. It's all correct. I can add weak acid, strong acid, weak base, strong base. But why are the pH solutions better? Because their pH remains constant. It does not change throughout the experiment. So they are more reliable. They're more accurate. Clear? Okay, so let's look at the picture. These are the two test tubes, no water bath here, and no equilibrium, and I will not leave it in the water bath for 10 minutes. One with the starch, one with the enzyme, with the buffer. The moment I mix them together, the moment I start my stopwatch, every minute I take one drop out, put it in the iodine until the color of the iodine remains yellow-brown. Does not change to blue. Clear? Excellent. Okay, this lesson, zero memorization. I don't want you to memorize anything. I want you to understand. What will we do now? Again, we will watch a video for this. In this video, it shows you nicely the...
I'm Professor Rick Mitchell, and I teach pathology at Victoria and also at Harvard Medical School. And I've seen that.
What is the role of the buffer? To change the pH and keep it constant. It doesn't change throughout the experiment. That's why it's active. Even they will put it in a water bath only to keep the temperature constant. The temperature will not change. And they will start the stopwatch once they mix it. Keep the temperature constant. This is the spotting tile. It's obvious here. They're adding a drop of iodine in every well. The iodine is yellow-brown in color, as you can see. This is the starting color of iodine. They will add one drop of pure starch to the well as a control. Use two to compare. Good. They just asked you. I'll wait again. Why did I wait again? Yes, the color is blue-black. This means starch is present or it has been broken down. It's still present. I'll continue again. Then I'll wait. Then I'll take another drop. This step is not important. I'll take another drop. I'll put it on the iodine. This time it did not change. So now starch is present or it is completely digested. Okay. Are the two experiments clear? Perfect. Good. Yes, my dear.
Yeah, start just to make sure that it starts
Just to compare that when starch is there, it will be blue black, and when it's not there, it will be yellow brown. To compare the colors.
Yeah, just to make sure it's not activates.
It's always starch, yes. Okay. The control here can be what he did, or you can boil the enzyme. It's always better to write boil the enzyme. I always see it in the mark scheme. Okay. Yes.
When you mixes the chemicals together, you have to wait for the reaction to start.
No, you start immediately, instantly. Yes, it's instant. The moment the enzyme touches the starch, immediately it will start breaking it down. Okay. Now, there is another graph. Is this touch? Oh, very good. Okay, there's another graph. Yes. Quickly, please. Now, this graph, the graph of pH and against the reaction rate. Again, we want to describe and we want to explain. Again, we will divide the graph into 3 parts. This is the easier graph. We'll finish it and we will be done. I have to use the laptop. Okay, I have to use the laptop. So here, again, we will divide the graph into 3 parts, A and B and C. B is the easiest part. What is B? Optimum pH. This is the pH where the enzyme is happiest. Okay? Now A, describe A. I will choose someone. Yes, a newcomer. What is happening at A? Describe. Yes, as the pH is increasing, what is happening to the rate of the reaction? Increases. Okay? Now, they will not ask you to explain. Because here, you cannot say anything about kinetic energy. You cannot say anything about temperature and speeding up of the molecules. Okay? What is happening is that enzymes, when they are at their optimum pH, they will work very well. But then when they are far away from their optimum pH, in other words, when they are here or here, when they are very extreme, far away from the optimum pH, they will denature. So the enzymes actually denature at this end of the graph. They denature here and they denature here. Remember, in temperature, in A, they don't denature. In temperature, they slow down. So enzymes at low temperature, they don't denature, they slow down. But in pH, it's different. They are working very well at B. Away from B, put in mind, away from B, away from the optimum pH, they denature. Now they denature here and they denature here. Now the question is why? Explain. Will you mention anything about kinetic energy?
No.
Will you mention anything about temperature and the speed of the molecules?
No.
Will you mention anything about the anything related to kinetic energy and speed? In pH? No. Then why does the enzyme denature when it is away from the optimum temperature? For one reason, because the bonds break. The extreme pH changes, breaks the bonds. When the bonds break, what will happen to the shape of the enzyme? It will change. What will happen to the shape of the active site? Change. So it's very easy, even when you come to study. Please sit properly. Excuse me. This is really important. When you come to study, always tell yourself, pH, any change from the optimum pH, the enzyme will denature. Why? Because the bonds break. Never mention anything about kinetic energy or speed when you talk about temperature, when you talk about pH. Never mention rate of collisions. Molecules, they move when the temperature increases because they gain kinetic energy. They move faster. They hit. All this when the temperature is increasing. It's got nothing to do with pH. When the pH is optimum, the enzyme is happy. When the pH is not optimum, the enzyme will die. Why? Because the bonds break. It's got nothing to do with the speed of the molecules or the kinetic energy or rate of collisions or whatever. You will say that here, let me erase again. At this point, the enzymes denature. And at this point, the enzymes denature when the pH is away from the optimum, because what happens to the bonds? The bonds break. The shape of the enzyme will change. The shape of the active site will change. Period. That's it. But when we are talking about temperature, only then do you mention kinetic energy and speed of the molecules and the rate of collisions. Do not mention anything about kinetic energy or speed of the molecules or the rate of collisions when you are discussing the effect of pH on enzyme action. Clear? This is the only part in the lesson that is supplement. So this is the part that they will ask you about in paper 4. The rest of the lesson is core. Now this part is here. Let's look at it together. This you will find it in year 10 notes, not in year 9 notes, because this is the only supplement part in the whole unit. At low temperature, as the temperature is increasing, and this part of the graph, and when the temperature is very high, at this part of the graph. At low temperature, there is little kinetic energy, less chances of successful collisions, less enzyme substrate complex, less product is produced. The reaction rate, when it is low, when the temperature is low, the enzymes are slow, but they're not denatured. When the temperature increases, the rate of the reaction will increase. Clear? As the temperature is increasing in this part of the graph, more kinetic energy, more successful collisions, more enzyme substrate, more product, the reaction is increasing. The molecules are moving faster. Fastest reaction rate is the optimum temperature. In the human body, the optimum temperature is 37. Easy? Very high temperatures, you will not say the enzymes are denatured and that's it. You have to explain what happens to the enzyme when it is denatured. The bonds break down, change in the shape of the enzyme, change in the shape of the active site. The active site will not be complementary. Give the examiner everything he wants. And I want you to study the name of 2 bonds. Very easy bonds, hydrogen bonds and peptide bonds. There is another bond called the ionic bond. You don't have to know anything else about them. Next year, what are these bonds? How do they bond? What is their structure? Everything next year. Just the names for this year. Clear? Okay, good. What else do I want to mention? Ooh. Okay, this is a very important table. Again, it is in year 10 notes because this is what you will say when you mention denaturing, whether in pH or high temperatures. The bonds break, the active site will change, the enzyme will no longer bind with the substrate, no enzyme substrate is formed, no products are produced, the reaction slows down gradually, not suddenly. It's a gradual process. That's why the graph is moving down. Put in mind. So today we said very important points. When you are discussing pH and why do the enzymes denature at different pH, don't mention what? Never mention, very good. Never mention kinetic energy or speed of molecules or successful collisions. Never mention anything about collisions at all. You only mention these in temperature. Either they increase or decrease. Clear? When you come to discuss that the enzyme is denatured, it's not enough to write denatured and go home. Because the bonds will break, because the shape will change, because the active site will not be complementary, because the enzyme will not be able to fit with the substrate and inform an enzyme substrate complex. Clear? What else did we say? Any reaction, this might come in the midterm, you never know. Any reaction that involves color, this is an error. Any investigation that uses a dropper, this is also an error because the drop sizes can be different. What else? Yes, you have to use a thermostatically controlled water bath. The starting time, they all have to be the same. Do you know now what's a pH solution? What's a pH buffer solution?
Yes.
Okay. On Tuesday, I will explain only one experiment or two, which is this one, the catalase enzyme. Yes, I already introduced you to it. I will just go over it and we will solve paper 6 Tuesday, all Tuesday. For this lesson, you will not solve paper 4, only paper 2. Because paper 4, we will solve it when we take digestion. Fine. What you will do now, get out paper 2. Don't look at me. Get out classified paper 2 and solve the questions on enzymes only and finish it in class because the other 2 classes, even though you are much smarter than them and they will not be happy when they hear that, they finished paper 2 in class. Then what? Which one? It's all in your notes. This one? Okay, do not solve. You will solve together, or we solve together or alone? Paper 2, yes, you can solve alone. Paper 6 on Tuesday, the whole block also will be for paper 6. Fine. Do also questions on digestion. If you don't have to solve them, fine. What? Desperate? What's your favorite?